Hello students. So today we are going to discuss about structure of neuron. Structure of neuron before small introduction about neuron. What neuron we are going to study here? It is a multipolar neuron. Multipolar neurons are type of neurons what receive impulses from many directions with the help of many dendrons, but they transmit impulse always in one direction, single direction. That is through single axon. So multiple dendron, single axon, this type of neuron is called multipolar neuron. One short introduction about neuron. Neuron is a specialized cell in our nervous system. What it is responsible for impulse conduction, impulse to receive, impulse to analyze, impulse to process. So it is made for impulse receiving, impulse processing and impulse conduction. Conduction means transmission to passing it to next area. That next area may be any neuron, that next area may be any muscle. So let's start here. In structure of neuron, our first area is cell body. The cell body area is the main central area, large area, and nucleus it will be at center. Nucleus always at center. This large nucleus you can see, it is always at center. Then the body of neuron, it is having multiple projections. These projections what going out, these are known as dendron. The word dendron means small tree-like structure. So here it is small tree-like structure known as dendron. Dendron, dendrite, you can use it as common. Dendron, dendrite are same. Dendrons, if I'll zoom here, you will see some tiny spot like structure. This tiny dot like structure, if visible, it is called, this tiny dot like structures if visible are known as, I'll highlight here, this tiny dot like structure are receptors or known as spike or receptors. These receptors, they receive neurotransmitter and they make one impulse here. This impulse propagates in neuron and goes to either next neuron or to next muscle. So coming back to the main structure, our neuron main cell body, it is having multiple dendron, it is having central nucleus, it is having this equal to sign-like structure what it is visible on your screen, that is known as neurofibrils. And these green dot-like structures are known as Nissl's granule. These equal to like structures are tubulin protein structure. These tubulin protein structures are neurofibrils. Neurofibrils. And these green dots, what you can see, it is called Nissl's granule. Nissl's granule is nothing but the rough endoplasmic reticulum fragments. Rough endoplasmic reticulum, tiny fragments responsible for protein synthesis. Multiple types of protein synthesis. Maybe cell repair protein, maybe neurotransmitter protein, but maximum neurotransmitters are synthesized in terminal area, this area. But sometimes neurotransmitters are synthesized in this area also. Okay, or you can say neurotransmitter precursor molecules. Okay, then these equal to sign structures, neurofibril structures, are absent in core dendron area, this side, but present in the base of dendron, then present in entire axon. This structure, long cylindrical structure, is known as axon. Axon, these neurofibrils, they help in impulse conduction, ionic conduction. So that we'll discuss in impulse conduction mechanism. Okay, how neuron it works. Now we are studying, now we are discussing about structure of neuron. So we'll omit that, we'll leave that. We'll discuss that in structure of, sorry, impulse conduction of in neuron. Now this part, the neck part of any neuron is known as axon hillock or trigger zone. Axon hillock or trigger zone. Here only the axon potential starts. When I'm saying axon potential means nerve impulse. So nerve impulse mainly it starts from here. That's why it is called spike initiation zone or axon hillock or It is also known as trigger zone. Trigger, here impulse, it is triggered. It moves in this direction, triggered. Then the whole structure of this axon, it is wrapped around some neuroglial cells. Some neuroglial cells, they wrap around it. They wrap it, they cover it. When they cover with continuous layers, it is called myelin sheath. When continuous layers are present, in this green part you can visualize, this green part is continuous layer of covering, continuous multiple layers of covering. Then it is called myelin sheath. This myelin sheath what made here, it can be from or it may be by either by Schwann cell, either by Schwann cells or by oligodendrocyte. If we are discussing peripheral nervous system, means spinal cord neuron. If we are discussing spinal cord neuron and brain neuron, then it will be oligodendrocyte. If we are discussing about peripheral nervous system, spinal nerve, cranial nerve, then it will be Schwann cell. If you are discussing about brain and spinal cord, if you are discussing about brain and spinal cord, then this Schwann cell, it will be oligodendrocyte. Yes. Then these cells, what you can see here with yellow boundary, this yellow boundary cell, these are Schwann cells. Green area is myelin sheath. So myelin sheath, Schwann cell, gap present in between two Schwann cells, myelin sheath area, gap present. This gap is called node of Ranvier. This gap is called node of Ranvier. Node of Ranvier is responsible for impulse jumping. Impulse used to jump from one node of Ranvier to next node of Ranvier. One node of Ranvier to next node of Ranvier. That we will discuss in upcoming classes when we will discuss about impulse conduction. So again, come back to this. This is axon. Axon is very long. Longest structure of neuron is axon. Axon mainly conducts the impulse. In this is Schwann cell, Schwann cell, Schwann cell, Schwann cell. Schwann cell gap is called node of Ranvier, and in node of Ranvier area, myelin sheath is absent. Myelin sheath is missing. It at node of Ranvier, you can see myelin sheath, green area is missing. Schwann cell, cell cytoplasm, Schwann cell, cytoplasmic membrane present here. But there is no myelin sheath, green area missing. This missing area where myelin sheath is missing, that is called node of Ranvier. Very important for fast impulse conduction. And if this covering, what I said, continuous multiple layer is not continuous multiple layer, if it is just enclosed like this, just covering, just holding, just Schwann cell, it is holding axon, then it is called unmyelinated neuron. When it is with multiple layers, it is called myelinated. When it is just holding, it is called enclosed condition. It is not myelinated, it is called unmyelinated. Unmyelinated neurons, you need to note it down, you need to understand this. Unmyelinated neurons, they have comparatively slow impulse conduction, and when neuron is having continuous myelination or myelin sheath present, node of Ranvier present, okay, and myelin sheath, it is properly wrapping the axon area, it will give faster impulse conduction compared to unmyelinated or non-myelinated. Non-myelinated neurons, yes, there'll be myelinated, myelin forming cells, but that will not be forming myelin. There'll be Schwann cell. But the condition where multiple layers of myelin sheaths are not present, we call it unmyelinated. So in case of unmyelinated, impulse conduction will be slow. In case of myelinated, impulse conduction will be fast comparatively. Exactly, unmyelinated is also fast. But when we say fast or slow, it is comparative word. Like I'll say that this pen is smaller than my hand. So compared to my hand, it is smaller. But as compared to my finger, yes, it is longer. So longer, smaller, bigger, larger, faster, slower. These are comparative words. Don't get confused. Yes, without myelin sheath also it is very fast. but the myelinated one is very fast compared to yes. Now come to next part. In axon area, you will see there is no Nissl's granule. So you need to remember this, that in axon area, no Nissl granules are absent. Then you come to this area, the terminal part of any neuron. Terminal part, end part. End part again, it is having some branching. So it is also looking like tree. So it is called telodendria. Dendron means we studied tree-like structure. This is also looking like tree-like structure. But it is terminal part, so called telodendria. Terminal, telodendria. So this telodendria, the end part, these areas having swollen nodes. These swollen nodes are called synaptic knob or synaptic bulb or sole foot. If it is attached to any muscle, generally we call it sole foot. If it is not attached to muscle, if it is attached to neuron, it is called telodendria, sole foot for muscle. Telodendria is whole structure. Sole foot word we use for this swollen structure for muscle attachment. If it is attached to neuron, if it is attached to suppose directly to any next neuron, we call it synaptic bulb or synaptic knob. Synaptic bulb, swollen, so bulb, and synaptic knob, both are same. Inside synaptic knob, you will see large number of synaptic vesicles are present. Here in the synaptic knobs, you will see large number of synaptic vesicles are present. Synaptic vesicles, they are loaded with neurotransmitters. Neurotransmitters may be excitatory, may be inhibitory. We'll discuss about that in impulse transmission, impulse conduction. So, neurotransmitters are filled in synaptic vesicle. Many synaptic vesicles are present in single synaptic knob, and synaptic knob many are present or attached to one single axon. Exactly, when impulse it is carried here, axon potential it is carried here, then it propagates into multiple direction and it is delivered to different neuron or maybe same neuron or maybe different muscle or same muscle. So this is the role axon of one neuron to propagate impulse in one direction, receiving from many directions by multiple dendron. Multiple dendron it is receiving and propagating in one direction. This is called multipolar neuron. Now coming to the remaining labeled areas of this diagram, Nissl's granule we discussed, spike proteins we discussed, receptor proteins, axon hillock. Axon hillock is the neck part of neuron. Then axon area we discussed, nucleus of Schwann cell. These are repeated Schwann cells. Schwann cells are neuroglial cells, supporting cells. They support neuron and loaded with nucleus. Yes, nucleus as cell is there, live cell, so nucleus present. Node of Renvier we discussed, telodendria we discussed. Then coming to lower side leveling, dendron word discussed, forms dendrite. Dendron is tree-like structure, small tree-like structure. Cyton, the central area, main area. Then we discuss about neurofibrils. Neurofibrils are for internal conduction. You have to remember this, internal conduction, NEET exam or any exam. Remember neurofibrils, internal conduction. Myelin sheath we discussed, and we discussed about synaptic bulb. Synaptic bulb, synaptic knob, this side, okay, it contains a huge number of neurotransmitters in synaptic vesicles. One synaptic bulb contains many synaptic vesicles. Okay, and then one last point here we are going to discuss is exolemma. Exolemma is this white membrane of this axon. Axon plasma membrane is known as exolemma. Exolemma. Okay, students, hope you understood this. Tata, bye bye, see you.